o'clock Eastern Time. Good morning, everyone. Good morning, good afternoon, good evening to all our colleagues around the world. My name is Dil Thavarajah. I'm a professor of pulse quality and nutritional breeding at the Plant and Environmental Sciences, Clemson University. I oversee the Organic Plant Breeding Institute, which is the part of the seminar series to educate our growers, scholars, researchers, consumers on organic agriculture, especially for pulse crop development and nutrition. Funding for this project is from our National Institute of Food and Agriculture, NIFA, USDA, OREI, and USAID, Feed the Future Innovation Lab, and USDA Agriculture Research Services, ARS, actually that is from Dr. Vandemark's program, and then the Good Food Institute and the South Carolina Department of Agriculture. Today, I welcome you all for our second fall seminar. This fall, we are inviting all global breeders around the world, and we had a last seminar from a lentil breeder, and this seminar we are listening from the globally recognized breeder, Dr. George Vandemark, for the chickpea breeding and management. A little bit about the speaker. I met Dr. Vandemark actually a decade ago in Turkey, Anatolia, in one of the lentil conferences. And since then, we have been working together for more than a decade. I have been helping him with his breeding program for the biofortification research. So Dr. Vandemark is a research leader, acting associate director for the Pacific West area right now, and also he's a research genetist with the USDA in Pullman, Washington, U.S.A. His research focuses on developing improved cultivars of chickpea that are best suitable for the regions of North Plains of Montana and North Dakota, Idaho, and the Washington regions. And he also a great big fan of biofortification. He has done quite a bit of work on protein biofortification, fatty acids, probiotic carbohydrate. He is especially interested in coupling increased yielding to enhance other traits, including the nutritional quality, like I said before, resistant to soil-borne diseases, and the other symbiotic association with beneficial rhizobia. So with that, I welcome Dr. George Vandemark into the floor. Thank you, Dr. Vandemark. We can hear and see very well. Thank you so much.
Good day, everyone. Thank you for your participation in this seminar series. And thanks very much to Dr. Dil Thavarajah for inviting me and giving me the chance to present. The only thing that would be nicer if I would have been able to travel to Clemson and give the seminar there. I'm looking forward to doing that someday. So I'm located in Pullman, Washington. My name's George Vandemark, and I'm the research leader of the USDA Grain Legume Genetics and Physiology Research Unit. We're located on the campus of Washington State University, along with 4 other USDA research units. Today, I'll be speaking about chickpea breeding and cultivar development. First, I'd like to start it off by just showing you all examples of what different chickpeas look like. And so if we go to 12 o'clock, you'll see that we've got a Desi type, a small microsperma chickpea. An example is a cultivar named Miles. And Desi chickpeas are very popular globally, but they're not produced commercially to an appreciable degree in the United States. And they're typically decorticated, which means the seed coats are removed, and they're used often for making stews such as dal. Then over to the right, we have small ones, small chickpeas. They're Kabuli type chickpeas. That example is called the billy bean. And these small chickpeas that are grown in the United States, they're typically processed and used for making hummus or other edible spreads. Then down at 6 o'clock, we have the most popular chickpea cultivar grown in the United States, developed by my predecessor, Dr. Fred Muehlbauer, and it's a Cafe Kabuli chickpea called Sierra. And the first thing you'll note is that it's lighter and larger than the billy beans, and that also with the desis, and it has a different shape than the desi types. And finally, over at 9 o'clock, we have what are called Spanish white chickpeas. An example grown in the United States is Troy, and these are large, very light colored chickpeas that are very high value and often used in salads. So there's three common cultivated chickpea plant types of the chickpea cultivated species, Cicer aureatino. And I'd like to show you those three plant types in closer detail. So the first one you'll see, there's what we call compound leaves that has small leaflets and purple flowers. And these are typically all desi chickpeas, wild relatives of chickpeas, and they tend to have small seeds. Then there's a type that's very popular in the United States. They have compound leaves and white flowers. And these are a lot of Kabuli chickpea cultivars grown in the US and throughout the world. And these include Nash, developed by the USDA, CDC Frontier, a high-yielding chickpea developed at the University of Saskatchewan, and billy beans, which are basically a local Pacific Northwest landrace that's very popular for producing hummus. And then the third type has simple leaves. You can see they're more lobate leaves, and they have white flowers. And these are, although they're not highly represented across the world, they're rather popular in the Pacific Northwest. And they include the most popular chickpea, Sierra, a close relative of Sierra, Sawyer, which is very high yielding, but a little bit small seed size. And then Royal, a chickpea we recently released that has large seed size and some drought tolerance. And here's an example of chickpea acres harvested in the United States over the course of approximately 30 years or so. And you can see that this data comes from NASS, the National Agricultural Statistics Service. And you can see that started taking records of chickpea production in about 1987. And then you can see at around 2004, they started to separate chickpeas in production according to whether they're large or small. And you'll note that according to NASS, large chickpeas are considered chickpeas that are larger than 7.9 millimeters, and small, of course, are less than them. And you'll see that there was a good increase in chickpea production in the United States pretty much until about 1916. And then we saw decreases that were likely somewhat associated with some tariffs with some of the most important importers of chickpea, including India. Now, you see small chickpeas and large chickpeas. And why am I bringing that up? And the reason is because a difference is paid in the United States, whether your chickpeas are large or small. And very typically, the premium on large chickpeas is somewhere on the order of 25 to 33% more payment than small chickpeas, although you can see around 2018, we did reach a point where large chickpeas had less value than small chickpeas, and that might have reflected our ability to move large chickpeas on an international market. Now, I'd like to note that just in the United States alone, there's at least 4 different ways of measuring chickpea size. And I'd like to show you down at the bottom 4 popular chickpeas in the United States. So we've got Billy beans, quite small, that's primarily used for edible spreads, CDC Frontier, Sierra, and Nash. And one way they're measured towards the left by NAS is to call them large, which means larger than 7.9 millimeters or 20/64ths of an inch. And when you look at them that way, Nash tends to produce approximately 95 to 98% large beans. Now, this is in the Pacific Northwest, mind you. Sierra, a little less. Billy bean does not produce an appreciable amount of large beans, irrespective of how you measure them. And where we see that is in what we call A beans. And A beans, that's a very popular way of rating beans in the Pacific Northwest. And A beans have to be larger than 22/64 inches or 8.7 millimeters in diameter. And when you evaluate these 3 cultivars, according to the A-bean criteria, again, you see that Nash typically produce somewhere in the 90% A-beans, Sierra in between 80s and 90s, and Billy beans between 0 and 5% A-beans. 100 seed weight is also a way of evaluating chickpea seed size that's very popular. And when you do it that way over the course of, these are results for 6-year averages in our breeding program, you'll see that 100 seed weight of Nash ranged from 57.2 to 59.5 grams, Sierra from 52 to 54.3 grams, and Billy beans from 30.4 to 32.6 grams per 100 seeds. And finally, some people like to refer to them in terms of seeds per ounce. And when you'll do it that way, you'll see that smaller numbers are indicative of larger seed size. And so there you can see often the largest Nashes might be considered a 48. Sierra in between a 52 and a 55, and Billy beans at times almost 100 seeds per ounce. So that's very important in terms of our chickpea trait priorities. In the US, our trait priorities include yield, but yield doesn't do very much for us in terms of high value chickpeas unless we also have good seed size and color. We have a couple diseases of importance we have to focus on, including ascochyta blight and seed and seedling by metalaxyl-resistant Pythium. Early maturity would be very nice because often we have to apply a desiccant herbicide before harvest so that we can properly harvest chickpeas in the Pacific Northwest. We grow peas, lentils, and chickpeas in the Pacific Northwest. And of those 3 crops, chickpea is absolutely the least cold tolerant, and we're getting towards northern edges of production zones for being able to produce large chickpeas. And we have a lot of aspect in the Pacific Northwest in our fields, a lot of slope, and that picture is an example of what you see all over the Palouse of a chickpea field, where parts of the fields, for example, those on the tops and along the edges of aspect tend to brown up and mature earlier than you can see the green areas that are following the movement of water down the field. certainly improved nutritional quality has emerged as a very important trait in the last decade. And also, I have an interest in niche varieties and markets. Um, and so, some requirements for variety development are identifying sources of genetic variation for the traits, generational advancement and selection for multiple traits, often simultaneously or serially, and multi-year replicated field trials. So, in terms of sources of genetic variation for chickpea, there are two major global sources of genetic variation for chickpea. A gene bank maintained by the International Crops Research Institute for the Semi-Arid Tropics, ICRISAT in Hyderabad, India, which maintains approximately 20,000 accessions. And the International Center for Agricultural Research in the Dry Areas, ICARTA in Beirut, Lebanon, that maintains approximately 15,000 accessions. And if anyone from either of those two great institutes are here, I beg your pardon if I misrepresented the actual number of accessions you have. These are numbers I got from a review I read recently. And much closer to me, in fact, in Pullman, Washington, is the USDA National Plant Germplasm System's Western Regional Plant Introduction Station. And they have 3,741 accessions. And so here's an example just showing some of the genetic diversity in terms of seed size, shape, and color that you can find in chickpeas simply within the collection contained by the Western Plant Regional Introduction Station. So you see both small, light chickpeas, you see chickpeas with several different colored seed coats, ranging from cream to black to green. So, a great source of genetic variation is using a chickpea diversity panel. And here's just a picture of what we've done growing up a chickpea diversity panel in the field in Washington State. These are small plots, but you can see there's tremendous variation, just that you can see from here in terms of plant stature, leaf type, degree of maturity. And so the ICRISAT chickpea mini core collection is a really globally useful and widely used diversity panel. And what this is it's an effort to survey variation in the entire collection using a reasonable number of samples. And they've developed, approximately 20,000 accessions in their collection. They developed a mini core collection with 211 accessions, approximately 1% of their total collection. And these were selected based on their origin and 22 traits that included traits such as 100 seed weight, number of pods per plant, plant height, flower color, seed coat color, days to mature. In this collection, there's 168 DESI types and 43 Kabuli types, which reflects the global importance of DESI chickpeas in terms of global nutrition. And you can see the geographic distribution of these entries, which also reflects the very much so, again, the relative importance of chickpea to nutrition in the different global geographic areas. So you can see not well represented terribly by American or European materials. Again, you can see some of that variation in this diversity panel. So this is a diversity panel that we use quite a bit, and we evaluated for many different traits, some of which I'll mention a little bit later in this presentation. We identify materials, we cross them with adapted materials, and then we conduct generational advancement and selection for multiple traits. And I thought I'd just mention a couple of these traits we work on. Very important factors impacting chickpea production in the US are disease, Chickpea bite, and Pythium seed rots. Of course, other diseases are important in other areas. For example, fusarium diseases have more importance in the northern plains of the US. Abiotic factors, cold and drought stress, are certainly important. And late maturity is an issue because of very often in commercial chickpea production, a desiccant herbicide is applied about 14 days before harvest to facilitate harvesting. There's an example of classic symptoms of Ascochyta blight on pods of chickpea. And I'd like to note that in about 2014, my colleague Wei-Dong Chen noted bare areas, which are included in those circles. I'm sorry they're not terribly evident, but growers were complaining about seeing areas in the field in Washington state where there wasn't good establishment in seeding. And so Dr. Chen went out to these fields and dug up chickpeas, and he saw a lot of rotted seeds and seedlings. Then brought these chickpeas to the lab and cultured them to find out what was growing on them. What he identified was a Pythium, and this Pythium was resistant to metalaxyl. And metalaxyl is a fungicide that's very commonly used to control Pythium diseases of chickpea. And here's an example of that assay where the upper panel, we have 2 Petri dishes of the resistant Pythium. On the left, it's grown on media that doesn't have metalaxyl. And on the right, it's grown in media with metalaxyl. And you can see that it grows very nicely on both medias. Whereas on the lower panels, there's a Pythium that's susceptible to metalaxyl. And you can see it grows quite well in the left, but its growth is very much inhibited on the right in the presence of metalaxyl. Now, this is important in the Pacific Northwest because typically our chickpeas, before they're planted for conventional planting, we apply a aggressive seed coating to try to manage some diseases and pests. And this seed coat includes mefenoxam, a fungicide that's used to control Pythium and Phytophthora, fludeoxonil, another fungicide that's used to control Fusarium and Rhizoctonia, thiabendazole, a fungicide used for Ascochyta blight management, the insecticide thiamethoxam for managing wireworms and seed pod maggot, sodium molybdenate, because molybdenum is an important cofactor needed for biological nitrogen fixation. We add the rhizobial inoculant. I beg your pardon, we could talk the entire time about the importance of biological nitrogen fixation in legume cultivation. Chickpeas, similar to peas and lentils, they promote colonization of beneficial rhizobacteria that fix nitrogen in their roots. And the most common chickpea symbiont is Mesorhizobium ciceri. And then we apply a dye. So you can see untreated chickpea seeds on the left and chickpea seeds on the right. So mefenoxam, which is an isomer of metalaxyl, that was rendered ineffective by these metalaxyl-resistant Pythiums. So what to do about this? Try to identify alternative seed treatments that control the disease and identify sources of resistance in chickpea and breed for resistance. And I'll briefly discuss this second point here. So we developed a bioassay that we can do in growth chambers on chickpea. Pythium is a disease of seed and seedlings. And so we have growth chamber assays where we either infest soil with the pathogen or we just have healthy soil that's uninfested in the pathogen. And on the left panel, what you see are pots that were planted exactly duplicate as the pots in the right. And you can see there's much less emergence of chickpea seeds in the inoculated pots, a result of the Pythium pathogen causing seed rot and seedling rot that impacts the ability of the seedling to germinate. And so what we wound up first doing was testing different amounts of disease pressure. Pythium produces an oospore. It's a long-lived survival spore. And so we selectively cultivated oospores of Pythium, and then we inoculated soil with different amounts, ranging from, on the left, the control, that there was no Pythium oospores, and then increasing the amounts from 25,000 oospores per pot all the way up to 200,000. We did these tests with 4 Czech cultivars, Miles, a Desi, and the 3 others are large Kabulis, Nash, a line, Flip 8492C, that's very popular for both breeding and genetic mapping, and Sierra. We had 5 seeds per pot, and of course, replicated pots. And what you're looking at are the means of multiple experiments. And you can see in the absence of disease with 5 seeds per pot, there's very little difference in the left column where Miles, Nash, Flip, and Sierra all had more than 4 seeds per pot emerge. But then as we start to add inoculum, you can see differences in their means, and you can see that Miles continues to produce significantly better germination than the 3 other classes until we reach the point of 200,000 oospores per pot. Then we've just swamped the ability of Miles to have some resistance against this pathogen. So we wound up ultimately using 25,000 oospores per pot. We've evaluated hundreds of materials for resistance to Pythium. And I'll just give you an example of some of the characteristics between resistant and susceptible materials. So I have the entry on the left, their resistant score out of possible 5, with higher scores being indicative of resistance, the market class, the grams per 100 seeds, and their seed coat. And basically what I like to note is that resistance very much tends to be associated with Desi chickpeas and small seed coats. That susceptibility tends to be associated with Kabuli chickpeas that typically have larger seed size and lighter seed coats. But that wasn't always the case because we also did identify some Desi chickpeas that have small seed sizes, but they had light seed coats and they were susceptible to disease. So we've developed an effective growth chamber assay. The Kabuli chickpeas appear to be very susceptible, and the Desi chickpeas appear to be more resistant. And this work was led by a grad student in our group and published recently, and there's the citation. Now I'd like to talk a little bit about ascochyta blight. It's certainly the most destructive disease of chickpea globally. There's at least 2 races of the pathogen in the United States. And we've screened this ICRASAT mini core collection for both races. And that 211 accession collection is about 75% DESI, 21% Kabuli, and 4% other. And we use this great mini dome assay developed by my colleague, Dr. Chen. And this mini dome assay, basically, it's a Dixie cup assay. My major professor is often so disparaging. He would call them Dixie cup assays, yet they can be quite effective. Where you grow the plants in medium, soil medium in a little Dixie cup, and you grow them up about 2 weeks. Then you inoculate them with a spray of ascochyta, and you cover that for a day in a growth chamber to create a humid environment. Then you remove that, allow disease to progress for 13 days or so, and evaluate disease resistance. We evaluate disease resistance using a scale of 1 to 9, where you can see how they look in terms of both in containers and also taken out of the containers. With typically, we consider 1 to 3 to be a solid resistant reaction, 4 to 6 to be an intermediate reaction, and 7 to 9 to be susceptible reactions. Ultimately, we take them out of the growth chamber into our greenhouse and grow out the materials that are promising based on their resistance. We did this with the ICRASAT materials, and you'll see we had distributions approximating normal distributions for both race 1 and race 2 reactions. And we identified 8 accessions that were resistant to both race 1 and race 2. And we've incorporated some of these into our breeding program. I also mentioned improving nutritional quality. This is a tremendously important trait. And I'd like to note that there are some challenges often that come with breeding for improved nutrition in terms of sources of desirable traits. Here's an example of 3 parents, Sierra, a highly desirable Kabuli chickpea, and then 2 plant introduction accessions we identified that were very high in mineral concentrations for iron and zinc. And iron and zinc are certainly our targets for mineral biofortification. They are the minerals of greatest global concern. But what I'd like you to note that both of those parents are much smaller than Sierra. They don't have the same seed size as Sierra. And the PI 36122 is a DESI type. And so when you make these crosses, there's definitely some linkage drag associated with that. And it takes time to identify materials that have commercial seed traits and also higher nutritional qualities. But that's an example of what we're working with. Similarly, I'm working a great deal with Dr. Thavarajah on improving protein concentration in chickpeas. And here's an example of 3 breeding lines that we use to cross to Sierra and also to 2 other varieties from our program, Sawyer and Royal, that are very well adapted to the Pacific Northwest. And so again, you can see that although these materials have excellent high elevated protein concentrations, they're not terribly desirable in terms of seed size or shape or likely yield, or dealing with trying to bring all those traits together. And I'd like to mention some older results on, you identify sources of variation for traits of interest, and that's very good. Another important prerequisite for breeding is understanding the relative contributions of genetic environment, and their interaction effects on important traits. And here's an example of some work done in the past where we looked at 22 elite Kabuli chickpea breeding lines or varieties. We evaluated them at 3 locations per year, and this was done over 2 years. And so this is a mean square analysis for mineral nutrient concentrations. And without having to go through all of them, I would like you to note that on top, we have all the interactions, the genotype, the location, which you can think of as the environment, the year, and then all the interaction effects. And if we simply look at iron and zinc, the ones of most global importance, you can see that, yes, there is a genetic effect, which is heartening in terms of being able to improve the trait through breeding. However, the year effect and the location effects have much greater magnitude than the genotype effects, indicating that in this elite material, these non-genetic factors were very important in mineral nutrient concentrations. Dr. Thavarajah and I worked together on evaluating prebiotic carbohydrate concentrations in another collection of elite Kabuli breeding lines and cultivars. In this case, we looked at 18 entries at 2 locations per year across 2 years. And now, of course, these prebiotic carbohydrates, they are sugars, they are sugar alcohols, they are raffinose-like compounds, they're sugar-like moieties that are metabolized by our gut bacteria, and they're associated with beneficial health effects ranging from reduced incidences of cardiovascular disease, possibly helping to reduce incidence of cancer and diabetes also. I would like you to note again some mean square analysis on this, and somewhat similar to the case of minerals, we found again that although we did detect significant genetic effects in several of these prebiotic carbohydrates, including fructose, sucrose, and raffinose, and find that the year and the environment by year effect were most important in conditioning this trait. And this was quite interesting. And what we observed was that the concentrations of these compounds were highest in both locations in years that were hotter and drier. And that's somewhat intuitive when we remember that a lot of these compounds are also implicated as osmoprotectants involved in salt and drought stress. Dr. Thavarajah and I are really aggressively working on trying to improve protein concentration in chickpea. That's clearly the prize. And I'll just mention briefly, we're trying to work out the data still on some analysis we did where we looked at 17 elite entries over 4 locations per year for 2 years. All I can say is at the moment that by analysis of variance, we detected significant entry effects, significant year effects, but not significant location or environment effects. And I still have to parse out the interaction effects. I comment that the grand mean of all these entries was 20.3% protein, and the range of entry means was from 17.5 to 22.6%. And we're working on a manuscript for this. And I'd be remiss if I didn't mention chickpea genomics in this presentation. I do only a very modest amount of work in chickpea genomics. There's other labs far more proficient and accomplished at it than mine. But please let me mention that chickpea is, of course, a diploid organism that has 8 chromosomes. The reference genome has been sequenced. I'll just refer you to the seminal manuscript on this by Rajiv Varshney. It's approximately 738 megabases in size, which, like many organisms, about 30,000 genes. And there are ample genetic resources, the most contemporary being SNPs, the single nucleotide polymorphisms, and marker trait associations. They've been detected for many traits, including disease resistance, seed traits, yield, and nutritional qualities. And I'll describe recently some work a grad student finished up in our lab on identifying markers associated with resistance to Pythium ultimum. And we used 2 approaches for this. We used that classic composite interval mapping, where we had a chickpea interspecific recombinant inbred line population. We had 177 recombinant inbred lines, and they were crossed between the cultivated chickpea, Cicer aureatinum, and the wild relative, Cicer reticulatum. We had genotype by sequencing done commercially on this material. And after filtering, we identified a little more than 1,000 SNPs across 8 linkage groups with a total genome coverage of 1,186 centimorgans, which was in line with other efforts to map this population. We're kind of happy in terms of identifying 2 significant QTLs for resistance to Pythium ultimum. One we're calling Q4.1 and the other Q8.1. Q4.1 is located on chromosome 4, and that's the one where we're really excited to having identified, because if you move over to the second column from the right, you can see that explained, the presence of that QTL explained almost 50% of the total variation for resistance to Pythium in this recombinant inbred line population. Then we also conducted genome-wide association studies where we used 184 entries out of this 211 accession ICRISAT mini core collection. We sent the student to ICRISAT in India, where they so graciously provided us access to all of the molecular marker data that we had. And the student was able to filter out over 302,000 SNPs that we used for GWAS. And here's an example of the QQ plots, which indicated the model we chose, and the Manhattan plots for identifying significant markers. And just to highlight the Manhattan plots, on the right, we've got that threshold line, horizontal line, and those markers that are above the threshold line were significantly associated with disease resistance by GWAS. And what I'd like to note that one marker identified on chromosome 4 by GWAS, that was also found inside the QTL that I just spoke about previously, that we identified in the recombinant inbred line. So we're pretty happy about validating the presence of the QTL. And similar to other groups, we identified several candidate genes that may be involved in disease resistance, including zinc finger proteins, AT hooks for DNA binding proteins, and genes involved in ethylene biosynthesis. And I'd like to just end by, of course, showing you our field programs. We do a massive amount of field evaluation. It's quite temperate in Pullman, Washington. So this is how things looked in my yield trials 21 days after planting in Pullman. Sorry, this is July 7th, later on in the year, and you can see we've got flowering going on and the canopy's starting to fill nicely. And then finally on July 30th, and I'd like to note here that although Desi's are not grown appreciably in the United States, I have an interest in trying to see if we can find a place for Desis in American agriculture. And this year was my first attempt to evaluate Desi chickpeas in replicated yield trials. And I planted the Kabulis right next to the Desis, and we've got 30 Desi chickpeas replicated 3 times, but only in one location. And I just took this picture down the middle of the row, middle of the field. So you can see that the Desi chickpeas in the Pacific Northwest do mature earlier than the Kabuli chickpeas. And all this work, of course, is done in the idea of releasing new varieties. And 2 varieties we recently released were Nash in 2014, and this variety was PVP'd. We obtained plant variety protection for it, and it's licensed to the Washington State Crop Improvement Association. And similarly, in 2021, we released USDA Quinn. Nash was released based on its high yield and large seed size. USDA Quinn was released based on exceptionally high yield and seed size similar to Sierra. And I'd like to just note the product pipeline continues. Here's a couple promising breeding lines of newer materials. We've got Sierra on the left, this 119C and 0057C. And these are efforts to produce higher yielding, larger chickpeas that also have lighter seed color. And these are in the product pipeline. And so I would like to say thank you very much to all of you for your participation, and especially to Dr. Thavarajah and for Clemson for giving me the chance to give this talk. And I would like to just note the cooperators in this work I've described. Look, Dr. Thavarajah's up top. Dr. Dil Thavarajah, one of just my most productive and pleasant cooperations. Dr. Wei Dung Chen, plant pathologist extraordinaire, Jared and Jeff, who are great research technicians for our program, Dr. Michael Grusak, the center director for the USDA in Fargo, North Dakota, Sindhu Sankaran, a professor at Washington State University, who's very instrumental in helping us apply remote sensing to our breeding program. Chickpea Agarwal, a recently graduated graduate student with our program. Cooperative extension scientists. We like our results, but we really believe in our materials. When Kurt Schroeder from the University of Idaho and Steve Van Vliet from Washington State University, when they test them independently in their statewide yield trials. Dr. Rajiv Varshni, the absolute leader in chickpea genomics and genetics globally at Murdoch University in Australia. Priyanka Joshi, FAO postdoc, who's at ICRASAT now. The U.S.A. Dry Pea and Lentil Council, our primary and most effective stakeholder group, Washington State Crop Improvement Association, and numerous grower cooperators. And now, if there's any time, I'd be happy to answer a question or two.
Absolutely. Thank you so much for wonderful descriptive presentation. This is what we want to know, because chickpea is a very questioning, most people question in the growers, even in South Carolina here, because it's a new crop. But I want to start with the bigger question, because you're the global leader of chickpea breeding. Dr. Vandemark, the climate is changing, right? And then the chickpea is very susceptible to all these diseases. And I'm sure you're experiencing this kind of weather in Paulus area as well. So with increasing temperature and increasing rainfall or decreasing or maybe increasing the cold weather, how do you manage your breeding program to changing climate?
So, thank you very much for that question. And so the question was, in general, how do we try to manage our breeding program in the face of climate variability? And I would say we primarily do that by evaluating our materials over multiple years and multiple locations. And we're constantly trying to expand areas where we are trying to see how our materials do. Certainly, if there is more spring rainfall and cooler spring temperatures, ascochyta blight can be more of a problem. And again, cool spring temperatures and moist soil are conducive to oomycete diseases, such as Pythium. Um, we find growers are the most growers often are the most willing to try, and growers in the Palouse have been constantly trying to increase the northern edge of chickpea production.
Right.
We see that also, of course, Montana and North Dakota have colder growing seasons. They're more of a challenge in general, I would say, for chickpea production. And they have some challenges in terms of being able to produce large seed there, for example. And so that's something that's quite important. We don't necessarily find that the materials perform the same in different environments.
Right, right. So that lead to a question like, we start growing chickpeas here in South Carolina as well as Nebraska and also in Florida. So we have a warmer winters. And last year we had ascochyta is just wiped out. Even they didn't grow up very well. And also we had pod borer. So I don't know whether you have those kind of issues and what kind of advice you give to South Carolina growers that to avoid these kind of problems. And this is our second year trying chickpeas, and hopefully we will try some of your varieties this year.
Yeah. Okay, this is where I mentioned niche varieties. Okay, I think you'll find that there is more resistance to ascochyta blight available in DESI materials.
Mm-hmm.
And I think, at least conventionally, it's important, of course, to be scouting your fields and applying a fungicide before diseases just overwhelmed your field.
Exactly.
And I didn't talk a lot about management, of course, here. I'd encourage anyone interested in some more management specifics to contact me directly, and I'll communicate with you and with Jeff and Jared, our research technicians, who really are the boots on the ground as far as management expertise. But generally speaking, I would suggest when you're trying to grow chickpeas in a new area, you might want to start trying to grow some of the small seeded varieties. right? So you don't have the pressure upon you to produce a certain seed size. And what you harvest, you may well be able to at least make into edible spreads. You might find local producers that would like to be involved in something like that. And I think as we develop pulse crops more as ingredients for food, it may be more possible to develop product using DESI chickpeas.
Right. Exactly. So actually, Dr. Carly Ardway is joining as well, and she's agreeing with you. And she also saying there are many fungicides on the market, and not all do a good job of suppressing ascochyta blight. Get the product on early as soon as lesions are observed, which we did. And if there is a rainfall, apply along a 10 to 14-day timeline. So that was the disaster for these warmer climates to having chickpea. But with yours and Audrey's help, we are moving forward for this year. There are a lot of questions, Dr. Vandemark. I have to keep you for a little bit, 5 minutes. Some of the questions are burning questions like, this is a question from Dr. Shai. Do you routinely use seed treatments in your field nurseries of early generation populations and evaluation of your advanced breeding lines?
Okay, that's a great question. The question was whether I routinely use seed treatments in early evaluations and in field trials. And the answer is yes. We do use seed treatments, and I didn't describe some of the earlier generation selections. And I know different breeding programs do things differently. I'm certainly no global leader in chickpea breeding. We've got a regional program where we try to address needs of the Pacific Northwest and national needs primarily. So typically we make our crosses in the greenhouse. The F1s we've tried both hydroponically and in large pots. I find I get better success just growing F1s in large pots in the greenhouse. Then everything from F2 and on is grown out in the field. Depending on the success of crossing, we either do single seed descent or we do modified bulk breeding programs. And typically the earlier generations from about F2 through F5, they're grown in the field in the form of paired rows, just 2 rows at a time. The F2s will grow as many paired rows as we have for the seed, but of course, the F3s, what we're doing is we're pulling individual F2s, harvesting all the seed off the F2s, and then planting that seed out as a paired row. And in those, yes, we do apply seed treatments.
Yes, sorry, Dr. Vandemark. Go ahead. There's one more question. Yeah, there's 2 more questions regarding, I will ask you 2 more questions. One is from a graduate student. Actually, she's a chickpea breeder from India, but she's a PhD student here in the US. And she's asking, are both asexual and sexual types of ascochyta present in the United States?
The question is, are there both asexual and sexual forms of ascochyta? Okay, so the teleomorph, the sexual form is Didymela pinoides, Didymela pisci. And I'm sorry, I do not know if that has been detected in the United States. I would suggest contacting Dr. Wei-Dong Chen directly for that answer. Dr. Chen is absolutely the global leader in ascochyta-blight diseases.
Yes, thank you so much, Dr. Vandemark. There are so many questions. What I'm going to do, I'm going to send you these questions so then later we can send these answers to quite a bit of people. But there are your students joining here. They want to say hello. And one student is Renuka Attanayaka.
Yes.
And then another student is Priyanka Joshi. And
hooray.
Wanted to say hello to you. And they have been texting and saying hi to you. And
well, hi to them. Okay. Two of my favorite scientists.
Right, they are your scientists. That's what they are. But there are a lot of management questions and the disease management question. Also, one last question was: is there any link between chickpea seed coat color and the nutrition?
That's a good question. Is there any link between seed coat color and nutrition? I'm sorry, I can't give a definitive answer for that. Certainly, the darker seed coats would be rich in polyphenols compared to the lighter seed coats, which may incur health benefits. And in terms of just use as a food ingredient, the Kabuli chickpeas are typically sold with their seed coats intact. They have thinner seed coats. The Desi chickpeas typically have thicker seed coats, and often for being used, they're decorticated, so that seed coat is removed. So by removing the seed coat, in the case of Desi chickpeas, you would be marginally increasing the protein concentration of the ingredient.
Absolutely. And I've seen that, and I can answer that question later. I can send the information to Craig. Yes, a lot of nutrition quality is high in desis than the Kabulis. So that's why some of the desis are used for the flour-based products, food products in India. And they have more carotenoids as well. But when you look at the starch composition, which is high in cafe type or the Kabuli types. So that's what Dr. Vandemark's breeding program has a range of nutritional traits based on the color as well, and also the taste. I hope you all enjoy today's seminar, and I wish we have more time to spend with Dr. Vandemark. Thank you so much for your generous knowledge and showing us everything about the breeding program, and everybody enjoyed it, I know. And I also hope that someone will eat, people who join here today, eat some chickpea hummus or some chickpea salad just to honor our webinar today for chickpeas. And thank you so much, Dr. Vandemark. And our next seminar would be one of the bean breeders, Dr. Matthew Blair. It will be November 16th. Hopefully, I will be not joining, and Nathan Johnson and Tristan Lawrence will be the ones hosting. And it will be great to hear about beans, and from lentils to chickpeas to beans. And thank you all for joining today. And I wish you for a great afternoon and a wonderful day. Thank you, Dr. Vandemark.
Thank you all. Take care. Thank you, Dr. Thavarajah. Good day, everyone.
Thank you.